Welcome to Module 2, Working with Data. This is the first lecture, Introduction to Tabular Data. Tabular data is very common in biological data analysis. This is a type of data that can be formatted in a table, so the data is arranged in rows and columns. This data can be completely numerical, or it can be categorical, or it can be a mixture. And common data formats include the comma-separated value format, CSV, or the tab-separated value format, TSV, where between each column is either a comma or a tab. If you're working with this type of data not in a coding scenario, common tools are Excel or Google Sheets. But today we're going to be talking about Python libraries for coding and analyzing tabular data, and common libraries are Pandas and NumPy. I've put an example of a small table data set here in the middle of the slide, where the top row is commonly referred to as the header. These are the names of the different columns. And the first column is commonly referred to as the index. These are the names of the rows. And then each of the individual cells are individual data points from the data set. We've already seen in Module 1 how many different biological data types can be captured or converted into tabular format. So some data types are always in tabular format, but some, such as microscopy data, bioimaging data, start out as imaging or video and then are converted into tabular format because it makes them easy to analyze. So some of the data types that we've seen before in this course that are captured in tabular format include the micro-CT measurements from the Open Science Data Repository dataset OSD 557, intraocular pressure from OSD 583, multiple types of microscopy staining that are converted into different types of numerical values, such as the average intensity or the total area or density. And we've also seen how RNA sequencing data, along with a lot of other genomics data, is commonly captured in a tabular format, with each value representing the number of copies of each gene that was being expressed in each sample. So in order to work with this type of data in Python, there are two very commonly used libraries, very powerful libraries that have different capabilities but work really well together. The first library is NumPy. So this is a common library for working with numerical data. NumPy is built around an array format or matrix format and provides many capabilities for numerical operations, mathematical functions, and is particularly powerful for statistical analysis of large numerical data sets. NumPy is known for its fast processing of large numerical data sets. Then alongside NumPy is the Pandas library. This is a well-known library for very flexible analysis of structured data. Unlike NumPy, Pandas makes it easy to work with data sets that are a mixture of numerical and categorical data. Pandas is built around a data frame format, which is how it captures its tabular data, and provides a lot of very easy to use and flexible capabilities for cleaning up data, aggregating, summarizing, and analyzing data. So in the notebook that accompanies this lecture, we're going to learn a few different ways to load data and explore it using Pandas. So a little bit of a preview here. Pandas provides a function called read CSV, which can easily read in a CSV or a TSV file with one line of code. You'll have to specify that the data is tab separated if it's a TSV. And the output of this function is a Pandas data frame. You can then use the .head function to view the first 5 rows or number of specified rows of a data frame. This is especially useful if your data set is very large and you just need to have a sneak preview, but you don't want to print the whole data frame out to the screen. Dataframe.info function is very useful to provide information about the data frame. How many values are in each column? What are the different data types? Since, as I mentioned, Pandas is good at dealing with both numerical and categorical, but you need to know sometimes what those different data types are in your data before moving forward. And then the .describe function is very useful because it provides a summary table of different mathematical calculations of the data frame, including mean and standard deviation. As I mentioned, pandas and NumPy work very well together. So mathematical operations in NumPy can use pandas data frames as input. So they don't need necessarily to use a NumPy array. So for example, NumPy.mean or NumPy.standard deviation, or STD, can calculate mean and standard deviation of a pandas data frame. And you specify whether you want to calculate each of these operations across each row or across each column. And you do that by passing in an axis parameter. So if we're talking about using tabular data for machine learning, one of the important aspects of preparing our data for a machine learning workflow is called feature scaling. This is an important pre-processing step before your data is ready for machine learning in many cases. Sometimes it's not appropriate, but in many cases it is. So feature scaling ensures that all features contribute equally to the model by bringing them to a common scale. And we're going to talk about a couple of different ways that we can do this. One of the reasons that we might want to perform feature scaling is that in larger data sets or complex data sets like biological data, RNA sequencing, there are features on much larger scales than other features. And so this prevents those larger values from dominating the machine learning process and skewing the results. This also improves our model interpretability because the impact of each feature is on a comparable scale. So we can directly compare each feature to each other. In many cases, this will be essential for distance-based algorithms like k-nearest neighbors or support vector machines. So I've put an example on the slide here of a data set where the stars show the different values before scaling. So you can see that all of these values are on very different scales. Some of them are very large, all the way up to 17 on the x-axis, 5 on the y-axis. But the y-axis and the x-axis are on very different scales. And then after scaling are the circles, where each of those data points exist approximately between 0 and 1 on both of the y-axis and the x-axis. So this is an example of scaling a widely diverse data set down to between 0 and 1. So as you can see, the relative relationships between each of those data points are preserved, but the actual values have been scaled down. So there are a couple of different types of feature scaling that are useful for numerical data that we'll be going through in the notebook. The first one on the left is called normalization, and this is what we just talked about in the example on the last slide, scaling the data using minimum and maximum values. So for example, minimum of 0, maximum of 1. In this case, data values are constrained to a specific range. And this is useful when the feature distribution is really not consistent across the data set. The calculation for normalization is quite simple. In order to get a scaled data point, you simply take the non-scaled data point, subtract the minimum value of that feature, and divide that by the maximum value of that feature minus the minimum value of that feature. Now on the right is another type of scaling, which is sometimes more appropriate than normalization. This is called standardization. And this scales the data using mean and standard deviation. So for example, it might scale the data so that it's centered over a mean of 0 with a standard deviation of 1. Here, data values are not constrained to a specific range. They're just constrained to the mean and standard deviation. And this is useful when feature distribution is more consistent. Again, this calculation is quite simple. In order to get a scaled data point for standardization, you simply take the non-scaled data point, subtract the mean, and divide by the standard deviation. So we'll be going over all of those aspects in the notebook. And these are some different ways that you can work with numerical, categorical, tabular data in biology when preparing for a machine learning workflow.